The human microbiome is this amazing collection of trillions of microorganisms that inhabit our body, both inside us and outside us. We are particularly interested in the gut microbiota, which are the microbes, that are bacteria, fungi, viruses that live inside our GI tract. One of the main ways that we interact with our environment is actually eating and drinking, and that passes through our GI tract. So our GI tract is actually very, very essential and very critical interface between outside world and our body. The gut microbiota sits exactly at that interface. So xenobiotics are any chemical compounds that are foreign to the human body, meaning that they are not part of our body naturally, but we encounter them through our environment.
Pesticides, nicotine, artificial sweeteners, pharmaceutical drugs. When these chemical compounds get in contact with the cells, they induce damage. This damage is called oxidative stress. And this stress induces or causes inflammation. And this inflammation, if we keep it for too long, it will lead to other health problems like diabetes, heart disease, and even cancer. So an example of this is that when we mix a common antidepressant with an artificial sweetener with the gut bacteria will reduce some substances that will damage the cells, and this damage will induce inflammation, and this will cause further health problems.
This is why it's very important to understand how xenobiotics like drugs and pollutants actually impact our gut microbiota and vice versa, so we can get a complete picture of our environment and human body interactions and how it actually affects the toxicity or efficacy of the compounds that you ingest.
I'm looking at how the toxins interact specifically with the mitochondria. So mitochondria, as you all know, is the powerhouse of the cell, but it's much more than that. It's sitting in the middle of all of the biochemical pathways that are working within your cell. These toxins can increase mitochondrial stress and cellular stress, and in turn cause cellular damage. The toxins we can look at are different types of pesticides. Then there are BPA and BPA-related compounds. These are chemicals that are used in making plastic products. And the other class of chemicals that I'm interested in are called PFASs, which are also known as forever chemicals. These chemicals are very hard to break down in nature.
So they can accumulate and that can cause problems. These chemicals are found in everyday items like non-stick cookware, raincoats, plastic containers, even in the coating of canned foods. So it's something that we cannot avoid. They're virtually everywhere and in every single person in the world.
Generally, most people wouldn't come across a high level of toxin in their day-to-day activities, but a low level of toxins we're always exposed to. So it's very important for us to figure out how toxic these compounds are so that we can better understand how to regulate these compounds in future.
So most approaches to understanding the safety of xenobiotics goes by one compound at a time. But this approach is very limited because we are exposed to tens of thousands of these compounds over our lifetime.
What sets our research apart from the other groups in the world is we are testing hundreds to thousands of compounds in different combinations in different cells from the body. Liver cells, gut cells, and even bacteria from the gut, trying to integrate more information.
So we generate large amount of data, which is an excellent platform to train computers to understand this data and pick up the patterns that we as human beings may not be able to pick it up because they are too complex.
Since we can't possibly look at these compounds individually by doing different experiments, the data that we generate in the lab is going to be able to inform what the machine learning or AI could do to understand how these compounds could be interacting with different proteins within the cell.
I think one great thing about MRC Toxicology Unit is that we do a lot of basic research, but we always have application in mind. There's a different joy making a real life impact. For example, one of my PhD students who just graduated, her PhD work has led to a startup. They are developing precision probiotics for detoxification of harmful xenobiotics. And I think this is a great example of what can be done here.
The more we understand, the better we understand how these compounds interact with each other, with our cells, with our bacteria in the gut, we can probably prevent or reduce the impact in our health.
Given that we are exposed to so many xenobiotics, and given that all of us are unique in terms of our microbiota, if we can discover patterns that can help us actually define the rules of toxicity and efficacy, I think that would be a great achievement.